for the opportunity to talk today to you. So it's going to be a hard task after this six very nice talk for me to do as well as they did, but also to avoid overlap because a lot have been said already. I'll try to. Don't get too scared if I swap some slide very quickly. It's just to avoid overlap. So at that time in the day, you might be all agreeing with me that there is a very strong relationship between nutrition, microbiota, and the immune system. And this interaction actually has been highly studied in the context of allergy. And one thing which is key in the context of microbiota is that, and I really like this statement, is that never before in human history have baby and children grown up so cleanly. And actually what these statements say behind is that we are becoming a very bad host to our microbe, which is really a combined things between the microbiota hypothesis and the hygiene hypothesis, leading to this increased non-communicable diseases. And that's going to be very important in the context of breastfeeding, of course, because since nutrition impacts microbiota and microbiota impacts the immune system, all of this being connected, breastfeeding is really one of the main things that should be studied in the context of baby and prevention of diseases. And it's very important to do things right from the beginning, like Valerie just mentioned earlier. There is a lot of influencer of microbiome and microbiota in general. And this actually has been described by numerous papers. This is just a review publication that shows some of them. And all of those have been linked more or less to either increased risk for disease, such as infection, or increased risk of allergy. We know that some of these influence can actually be modulated by breast milk already. And one of them, for example, is human milk oligosaccharides. So, and this is an example I like a lot, looking at here a cohort where you have babies which are born from C-section and baby which are vaginally delivered. And you actually see very nicely how actually the C-section born have a very different microbiota compared to the one which are vaginally born in blue. Here, the population actually have been also separated in subgroup, which are the ones which are from the mother which are able to do 2FL, so 2-fucosylactose, and the ones that were not able to actually produce that in breast milk. And you see very nicely that actually the orange population here is way closer to the blue one, which were born vaginally. And what it shows that is that specific human milk oligosaccharide, like here 2FL, in the mother milk can actually alleviate in part the effect that C-section could have on the gut microbiota. So human milk oligosaccharide is, of course, a big part of breast milk, but I'm not going to go into too much detail on structure. It will be talked about in 2 days. Here, I just wanted to say that it has been extremely difficult in the years before to actually study the beneficial effects that this human milk oligosaccharide could have on health, because we were not actually able to do intervention study. This is one which was done very recently that kind of confirmed what has been shown in a lot of observation study, that there is a trend that, there is a trend toward a protection when you actually supplement the babies with fucosylactose. The second example is really similar. In this example, the baby were also supplemented with 2-fucosylactose, and you can see that in this baby, actually, the supplementation was favoring less use of antibiotics, but also a lower risk for respiratory infection, specific respiratory infection. This actually has been also shown in numerous preclinical data. And here it's just 2 examples. One is a vaccine model it's a flu vaccine model in mice. And here you can see, and I will go back to it later, a very nice actually dose response of the supplementation with the HMOs. But what's interesting is actually there is an optimum dose. And then if you put too much, actually you lose some of the efficacy of the HMOs. And here it's another example, which is done here in the model where we actually infect mice with flu, adapted for murine species. And actually you see that these HMOs were actually able to protect the mice against the symptoms which are associated with infection. So we have to keep in mind that the maternal food 2 and food 3, meaning like the possibilities that the mother has to actually express a milk that contains 2 fucosylactose or 3 fucosylactose, is actually linked to the genetic of the mother. And that's quite important because the genetic of the mother predicts what will be the quantity of fucosyl in the milk. And it's always very important to actually look at subgroup in the mothers in the breast milk that can secrete the fucosyl lactose and the ones that cannot, to actually understand better what is associated with what benefits. And that's what we did in a cohort from Dhaka, and the slide was provided to me by Aristea Binya. And what we actually have found in this cohort is that actually the mother that was expressing fucosyl lactose, so that were put to positive, were actually having babies that were protected at 6 months against respiratory infection. And that's quite interesting when you look at that at 6 months, knowing that a lot of these babies were actually exclusively breastfed up to 6 months. But what's even more important is actually to look also at the infant, because if the mother is actually non-secretor, there is a chance, or secretor, there is a chance that the baby will carry the gene of secretor and non-secretor too. And we have to keep in mind that we actually have in the mucus layer some motif of glycosylation that really also bears this 2 fucosyl group. And actually, when we segregate the population by the genetic of the infant, here we see some quite interesting results that actually show that at 4 months of life, the maternal and infant food 2, food 3 status influences differently the protection against infection. And you see clearly here that actually in this group, in the infant which are negative compared to the infant which are positive, you have this inverted protection, which is certainly driven by the genetic of the infant rather than the breast milk. So my message here is really to keep in mind that we really look only things in silo most of the time, even in breast milk. And we are actually misled by not necessarily knowing all the confounder or studying all the confounders that can be associated either with the benefit or the actually level that we are looking at. It's just to summarize that a lot of things has been shown on the role and function of HMO. I'm not going to go through it. It will certainly be taken care of in 2 days. I'm just going to really stretch on the fact that we need to consider also the host genetic of the mother and of the kids when we actually look at this association study. What I'm really interested in the context of breast milk is actually allergy. And we just discussed earlier that allergen introduction is quite tricky. And in the context of breast milk, on the top of it, I mean, should we exclusive breastfeed till 6 months and avoid early introduction? Should we introduce, like Valérie said, very early, like the new LEAP study tend to mention that we should start at 4 months? Yeah, it's created a lot of confusion, and we really need to study more how and when to introduce this allergen, of course. But historically, I mean, we have been avoiding allergens for actually pregnant mother and during lactation. And that was actually certainly misled by the fact that if breastfed baby actually are allergic to human breast milk that contain allergen to which they are allergic, I mean, you need avoidance for sure. Now, what I will talk later in my talk is not necessarily preventing the symptoms of allergy, but really preventing the sensitization itself to the allergen. And why it's important to look at it, of course, it's because, I mean, allergy, and that's Australian data, and I think egg allergy is 9% in the infant population in terms of prevalence, so it's huge. in Australia. And you see that it's still increasing. We don't know when it's going to plateau, but estimates say that in a few years, 50% of the Australian population will be allergic. So it's quite a very important topic to tackle. Of course, and Valerie mentioned that earlier, allergy, the immune system, and the microbiota has been highly linked together in the literature. And here you have one example showing that actually, if you have a germ-free mice, it's actually have an underdeveloped immune system because it actually waits for the microbiota to get stimulated and efficient. This I would say, absence of exposure to the microbiota also leads to immune dysfunction, I would say, in these germ-free mice. And one of them is, of course, the elevated level of serum IgE antibody in this murine model, which are devoid of microbiota. So as far as HMO in breast milk and allergy, that's what we actually tried to study during the last years. And this is one study that I've shown that actually, if you look at the different composition of 2-fucosyl lactose in breast milk based on if the mother can secrete it or not secrete it, you actually would find that you have some kind of protection of being a secretor mother, but only in the babies that were C-section born. And that actually looped back very well to what I was presenting earlier with the effect of 2FL can have on the microbiota, alleviating this difference between C-section born infant and vaginally delivered infant. So we have actually tried in the last year to study deeply human milk in the context of allergy. And we were lucky enough to be able to access to two cohorts, one that we consider as an exploratory cohort, and one that we wanted to use as a confirmatory cohort. And any finding we will find in the first one, we will actually check in the second one how the finding shows. And these two cohorts were actually the live child cohort in Germany in collaboration with Leipzig University, and one in Thailand, where we also obtained breast milk. Both of these cohorts, I will show you later, have advantages and limitations, of course. But on the right of this slide, you have all the different components we analyzed. It's only part of it, but we had more than 50. And the goal was really to go more in I would say, multivariate kind of analysis to actually try to find the interaction that breast milk component could have in regard to allergy. So the first cohort is a German cohort, and we actually had 156 mother-infant pairs. We had 3 points of collection at 3 months, 6 months, and 12 months, where you see the inclusion decrease, of course, over time. We actually there quantify more than 25 HMOs, and we actually had multiple other components. We quantified allergen also in this breast milk component, and total Ig in the mother and the kid. The Thailandese cohort here was extremely well. So the German cohort, it was based on questionnaire to the parents, the way that allergy was tackled. So the parents were asked, is your infant clinically diagnosed with allergy, food allergy, eczema, etc. So, but it was really parent reporting. What was nice in this Thai cohort is that it was actually clinically diagnosed, and a very well allergy work was done in this population. The limitation we had here is actually the time of collection was actually very different. Some were collected very early on in the first month, and some were collected after 1 year, even almost 2. So we had a very large distribution in the collections, which create a big bias because component in breast milk varies with time. And we perform analysis. So here I just show you correlation matrix. You will get no key message out of it, except that it's very complex to analyze. because you find all these red dots, blue dots, saying it's positively correlated, negatively correlated, and you can look at each dot and actually find some sense out of it. And here you have TGF-beta1, TGF-beta2, IgA. I mean, the literature have shown before that those were more or less correlated together in breast milk. So some you can really find a rationale behind them, and some it's actually way more difficult to find something. So we actually ask a very smart statistician to actually work the data in a different way and to actually find, based on this correlation and a lot of statistic work behind, to find how the breast milk components were actually interacting one with the other. And what you see here is actually a plot which is generating in silico based on the level we have in breast milk. And each individual component is actually linked to all the other one. And then the computer asks, is it the best way to link these two components, or is it a better way? If there is a better way, the line is removed. And here you actually end up with the lines that explain best the correlation you have. And I think it's a very interesting, it was used in social networking, in political and economical field, but never necessarily in biology. And it actually creates a very nice tool to create new hypotheses around breast milk composition. And it's also, for me, very nice to see how complex breast milk interaction is, because it's not one component that you look at in relation to allergy. It's actually a mix, very complex, of a lot of components that interact all together. And if one is moving, it's certainly that a lot other around are actually going in maybe opposite or similar direction. From this analysis, we were actually also wanted to do univariate analysis. So the regular analysis where we take each component and we look how associated it is with allergy. We were not starting from scratch. Some publications were out there, and this is one of them, showing actually that, for example, NLFP3 is associated with cow's milk allergy. And NLFP3 levels, you see here, is associated with cow's milk allergy. And we also had another paper that was actually showing that if you do univariate analysis and you look at children's sensitization, actually there is nothing necessarily that you will find. But what the paper say in between the lines, there is one sentence that's actually lower level of 2FL were actually associated with prevention of allergy, which tell us two things. First, the association between allergy and HMO might not be that linear. And the second thing is that maybe there is still association if you dig a little bit further into the data. And if you remember early on, I was presenting a slide on infection, and I was showing a very nice, actually, bell-shaped reaction, showing that you have an effective dose, which is protecting, and the rest, actually, if you put too much, you actually lose the protection. And actually, when we digged into our data, is something similar that we actually found. And we wanted to test that sorry, I'll just go back here. So before that, testing it, I just wanted to say that we also look at the genetic of the infant and how that would actually influence allergy in the offspring. And we actually identified that some SNP, here again, where in the infant, in the cluster of FUT3 gene, was associated with some kind of risk to develop allergy. And this is a gene, SNP in FUT3. And the interesting thing is that when you have a mutation in this FUT3, you actually have an increased level of 2FL because you don't make FUT3 anymore, and you have this increased level of 2FL, which is a compensatory mechanism. So here we wanted really to see, is it the genetic, or is it really maybe the gene, the 2FL that is protective? So we test in mice, in a mice model, and here we use skin sensitization that we discussed earlier as a model to actually test our hypotheses. And what we found, and I will just show you the data on sensitization, is that actually the mix of HMO were able to, at 1%, were able to protect against allergic sensitization. However, the beneficial effect was lost at the higher doses. So we really need to actually be very smart when we look at such complex ingredients, because ultimately it doesn't function like a regular probiotic. The more you put, the better it is. In mice, actually, that's usually what we see with prebiotics. But here, we actually have a bell-shaped form. And that was actually troubling us because we knew from prior data that we did a few years back is that propionate, for example, when you feed a mice with propionate or when you feed a mice with fibers, you actually protect against sensitization and also lung inflammation. So basically, the two data were not fitting together. And we check what was going on in the context of microbiota and short-chain fatty acid production. And here, the higher dose still overproduces propionate. So we don't have a drop of propionate with a higher dose, but we do have a drop in sensitization, which we are actually not necessarily expecting. So we looked at other data out there, and we found that in the context of food allergy, HMO has already been tested, but it was a model where they were using intraperitoneal sensitization. So we said, and it was treatment, not prevention. So we said, okay, maybe we can actually look how oral sensitization this time would be prevented by HMO exposure. And here we were thinking that maybe in this model we will have a dose-response effect of the HMO and we will not lose the effect as higher dose. And what we found actually is actually very similar schematic, where actually we lose the effect with a higher dose. So it's very interesting for us to see that actually the FOXP3 regulatory cells were actually following the protection we were seeing with the sensitization. But it really tells us that ultimately it doesn't work the same on every system. And here it's another system that we look at. It's actually MCPT1 detection in the serum. And here you see that actually it's going down on a linear basis. So we have this very surprising interaction with the immune system with these HMOs that we are still trying to understand how it's working. But today my key message that I want to convey is that we always need, in the context of breast milk, to see maybe a larger picture, to look at the host, and try to find all the confounders that could really interfere with the systems. We have seen that HMO, such as 2FL or LNT, can decrease actually allergic sensitization, that regulatory T cell might be associated with this protection. But however, we didn't find any effect on lung inflammation, which is very surprising if it was just a prebiotic effect. So there is something more, something very specific about HMO, certainly, that we don't understand yet, and we are working on it. But yeah, I think it's very interesting to understand that what we see with HMO is not a simple prebiotic effect, and there is certainly direct and indirect effect on the immune system that we don't understand really well. And here is my acknowledgement slide. A lot of people are involved in the study I showed, some collaborator in Leipzig for the access to the milk samples, the infection and airway disease were from cohort obtained from International Center for Diarrheal Disease Research in Bangladesh, and the Thailand cohort was obtained from Chulalongkorn University. Thank you very much for your attention.
Thank you so much, Carine, really. We have some time, quite a bit of time for questions. Yes.
Very nice presentation. Is there any data on, I go back to the cow's milk allergy children, is there any data on HMO composition in breast milk of mothers of infants who develop cow milk allergy?
Yes, so there is one study that I showed, but I think that's the only one in human, that shows that there is 3 HMOs that stand out as protective for cow's milk allergy. And one in particular, which is standing still after confounding factors.
I missed that. For example, do children who are exclusively breastfed, however, they develop cow's milk allergy, does that HMO composition differ from other kids who might be at risk but do not develop cow milk allergy?
So just for HMO, that's the
Yeah.
only data is the study I showed, and that's the only one out there, showing a little bit lower level of LNFP3 in the children that were allergic to cow's milk.
So the observation is right. The mechanism is really something we would like to find out. You mentioned that regulated T cells may be important. Do you think the mucosal invariant T cells, which have been implicated with a variety of other disease processes, have you looked at them?
No, we didn't look at them. We actually today only looked at regulatory T cells. We were actually hoping, like what we usually see with this prebiotic effect, an increase in Treg at every dose. We're very, very, very dose dependent, but we didn't look at anything else yet.
You have not, okay. And what were the markers for allergy, symptoms you mentioned basically? Were any immunologic markers for allergy which you looked at?
So in the skin model, we looked at a lot of clinical symptoms. So we have the skin score, we have transepithelial water loss, for example. We have mast cell infiltration in the skin. We have so we looked at a lot of things, and we are really trying to put the puzzle all together to see what components, what compartment of the mouse react differently to the different to the same intervention ultimately, because it's a model where we can have the lung, the skin, the gut inflammation. So it allows us to actually have a very nice model to dig out the differences in terms of targeting or organ targeted for the protection. And it seems there is a lot of discrepancy compared to classical results that we would obtain in this model. So it's quite interesting.
Bo, you have any questions? No? Yes.
Yes, Frank Rimmel from Paris. Thank you for the wonderful presentation. If I understood well from the studies you showed, in humans you only have data on systemic sampling. You don't have access to the mucosal department, I think, particularly to the GI intestinal mucosa. In animal models, you have access, as you just highlighted, to understand the molecular mechanisms to all compartments. So do you think that your animal models reflect the allergy or atopic risk that you see in humans with regards to the polymorphisms for the focalization and also for the filagreen, as we just discussed before? Do you have the right model to test your hypothesis?
So it depends what the hypothesis was. The hypothesis was, can we decrease sensitization with HMO in an allergic model? We tested two, we can decrease the sensitization. So whether it's skin sensitization or gut sensitization, it does work. So I think for this, yes, we had the only model actually you can have. And we tested them. Now, of course, they are not deficient for filagreen. The one we put a patch, it's actually to impair the barrier and force sensitization. So it's a forced sensitization in mice. All the mice have the same genotype. We still have, outliers in our model. So, but I think it's more, it's part of modeling diseases and modeling nutrition. It's quite complex, and we can only have a little piece of information now as far as immunology is concerned. Rodents, and she was mentioning same reflection for ova. Rodent is quite well recognized for the immune system. Now for atopic dermatitis or for milk allergy and the clear clinical symptoms, can we translate that in human? We don't know. We will have to do intervention study to answer, of course.
Any other questions, comments? Yes.
Ferdinand Haschke. At the beginning, you showed two studies, clinical studies. Both were safety studies. The Puccio study, you mentioned the infection data, secondary outcomes.
Correct.
You didn't mention it at the Heidi Storm study. This was a safety study.
Correct.
And you showed briefly infection data. Were they similar, like in the Puccio cohort?
Similar in terms of prevalence or similar in terms of...
In terms of prevention, in Puccio had certain associations with lower prevalence of?
Yeah, I don't think there were subgroups similarly. It might be because the answer was different. I don't know. I will have to check
but
back.
But you showed the slide
yeah,
on
I
this.
can show back. Up. One more. Here. So in this one, it's 23% versus 8, and this one shows the odd ratio. So it's difficult to 1 to.
So not this I know, but the previous chart which you showed.
This one in partially infant, partially hydrolyzed formula.
Yeah, these were secondary outcomes.
Yeah,
of
it
course.
was. Yeah, of course.
Okay. Yes, Olaf?
Very interesting. Thank you very much. Do you want to comment at all about the variability in human milk oligosaccharides, given that probably is the most variable species we know of, and complicates matters of interpretation, perhaps?
For sure. So there is studies showing that there is a lot of geographical differences in HMO composition. The fucosyl one, of course, are part of them. It's always important to look also at the genetic, because genetic is not, the repartition of these SNPs are not equal according to the geography. And even the ones that compare urban versus rural parts and showing differences in HMO composition, here we are not controlled for genetic of the mother. So it's always very difficult to know what comes from the environment, what comes from the genetics. But yes, there is a lot of variation across geography, but also across timing when you actually collect the milk. And that was a big issue we had here in the Thai cohort, where we had collection at different time points, because some actually fall down drastically in terms of concentration between, the first week and a year of age. So, yeah, I mean, variability is huge. It's hidden, but I have a dot plot I can show you. I mean, with the repartition, which go from 0 when you are a non-secretor to 5,000, so 5 grams per liter. So, and you have the whole spectrum in between, with average. For example, in the Thai cohort, 2FL was average at 1.3 gram, and in the German cohort, it was 1.9. In median, sorry, it's median, 1.3 and 1.9. So, yeah, the median already, there is quite a lot of diversity between these two cohorts. So, yeah, variability is quite impressive, but it's quite interesting to understand what is really necessary and coming from the adaptation, and we need it for our survival somehow with a year and generation of evolution, and what is actually luxury.
Yeah, and I'll add to that just that humans are the most variable species in terms of HMOs of any mammal that we know of. So it's, and as you pointed out, there are differences among individual secretors and so forth and so on, and so it would suggest that there must be some reason for the variability, whatever it might be.
I agree. I don't know.
Okay, I think with that we close the session for the immunology of milk and lactation. And Natalia, would you like to close the session?